nervous system that we know a fair amount about, to have the chutzpah to think that we can really create meaningful models at this level of
neurobiological.
It absolutely is. The chutzpah, yeah, the chutzpah neurons. I have a decent number of those neurons. Who knows? But surely we have to try. So I'm going to tell you about birds, singing birds, and birdsong learning. And some of you, maybe many of you, don't really know much about it, but you've heard a bird sing, surely. And this is an amazing system. And you're going to learn a lot about the biology and the neurobiology of it. And it's going to set, and the organization of the brain, and it's going to set up a problem. Okay, and having, if I do a good job, if I get to the point of having it set it up well, then you will see the basis, the neuroscience basis of what we want to do with now taking these powerful techniques, as Henry has developed over a number of years, applied to the nervous system and creating these interesting models. Okay, so that's the agenda for this talk. So there are lots of different birds, right? And the thing that we like about songbirds, singing birds, is they have pretty songs. And the songs are different across species. That was a zebra finch. There are 5,000 species of songbirds, and 4,999 have beautiful songs. And there's the zebra finch also. That's a white-crowned sparrow. This is my thesis bird.
This is from the Tokyo subway.
This is an indigo bunting, which is a bird in the prairies of the United States and Canada. This is a bird you're familiar with. It's a canary, right? And this is now going to be 30 seconds. I'm going to play maybe all 30 seconds of the song of the European starling. So I don't know if you heard that. That's a very complex song, right? And this bird has a variety of different elements to its song. So there's a lot of variation across different species, and it has biological significance. So they're signaling to other animals when they're singing. And we can actually understand that variation, at least in part, in terms of the structure of the syrinx, the vocal organ, which is different from the mammalian larynx. The syrinx sits at the base of the trachea. It is most complicated, has the most number of muscles in the songbirds. It has variation that changes depending if you look at non-songbirds or a group of birds that are ancestral to true songbirds called neotropical songbirds. And there's also a lot of variation in the syringes within songbirds, or Aucine passerine birds. So a number of years ago, a colleague, a scientist in University of Buenos Aires, created a biophysical, a bioacoustical model of a nonlinear dynamical systems model of the action of the syrinx. And here you can see he started out with the physics of the movement, understanding that the membranes in the syrinx move in a flapping motion if there's sufficient pressure from that's driving air through the syrinx, and that movement, that flapping motion, then transmits fluctuations in the airflow into the column of air, which is where the sound which is how the sound is generated originally. Gabo and his colleagues did a an analysis of the dynamics of this model, which is dominated by a Hopf bifurcation and a saddle node and limit cycle bifurcation. I'm confident there are people in the audience here who can tell you much more about that than I do, but I've learned something about it over the years. And then a number of years later, they realized that the equations that they were dealing with could be moved in the parameter space, translated, stretched, and then they could be described by a standard set, or a so-called normal set of equations, normal form equations that had already been described for this type of dynamical system. And it's actually the same one that describes the production of sound in the larynx. So it's a very interesting connection between the two. So you have this highly nonlinear behavior of the syrinx that is then modified by resonances in the trachea and in the structure at the top of the trachea with a long, complicated name. We'll just call it the OEC. There's some other more recent additions to this model, and ultimately the bird produces a sound. Okay. This is going to be fairly low amplitude, so listen carefully. This is now the original song. Could you hear that at all? And that's the model song. And at such low amplitude, you can't hear the difference whatsoever. I apologize if you couldn't hear it, but the point is that this is doing a pretty good job of creating a synthetic song that is very similar to the original song. Gabo's hypothesis is that simple instructions, not complex instructions, but simple instructions from the brain are driving singing behavior, and most of the nonlinearity of the sound production is built into the syrinx here. And there's some evidence for that, but that's not my point here. My point here is simply to say that if we're now going to start building network models of the bird song system, which we'll talk about in a moment, we have a pretty good quantitative model that we can use to hook up our neural network models to. Right? So that's a nice starting point. So this is, there are lots of connections. We don't need to worry about most of them. We're going to not even talk about the auditory side of it. We're going to stay focused on the so-called song system side of it, the part that is actually producing the singing behavior, and where plasticity takes place. Well, plasticity takes place everywhere, but there are changes in the song system that's related to song learning. I'll tell you about that in a moment. And we're going to pay attention to three parts of the brain. One is called HVC, for reasons that are fading into the dim, distant past. We'll just call it HVC. The other is called RA, and the third is this pathway here, which we'll call the AFP, anterior forebrain pathway. So HVC is analogous to secondary or perhaps motor association cortex in mammals. RA is similar to the primary motor cortex. And this AFP, or basal ganglia loop, exists inside of your brain, to be sure, as well. And it's part of a general mammalian pattern. You'll see it produces different effects. So our job in general is going to be to understand the relationship between these three structures as a beginning for creating models, computational models for how the bird does what it does. Okay? So what is it that the bird does? Well, the first thing we need to think about is this diagram I showed you is kind of a top-down view of things. It's the standard way we've been thinking about it in neuroscience for a very long time. And it's almost certainly wrong. At least some of us believe that it has, that's a fair way to put it, some of us believe that it has significant limitations. You can also just recast those connections into a sort of bottom-up perspective, so that not only there are signals going from higher levels of the nervous system to lower levels that are driving the output, but there's also signals coming back from brainstem structures up to those higher levels, and they're very important for organizing the temporal structure of the behavior. Now, that's pretty interesting, right? Because That's a big fundamental difference in how you think about it, and we don't have the answer. Okay? So a model could help us address such a fundamental problem. And the way you think about the timing, the way you think about what is the master and what is the slave, what's driving what, and so on, matters depending on whether you have how the system is actually organized in these two different schemes. Okay, so let's look a little bit more at this. We have different parts of the brain. We have a connection between the different parts of the brain, and we're going to learn something about the organization of it. We're going to learn something both about the anatomy of it and the physiology of it, and we're going to relate that to the behavior. That's our goal here. And knowing all that will give us the substrate, the basis for making biologically interesting models. Okay, that's our program here. So remember I told you about topographic organization in the electric fish? Well, in this motor system, there's also topography. So this is a motor system because these structures are involved in production of the song. Right? So it's motor in that critical sense, that fundamental sense. You can now put injections of tracer chemicals into different muscles of the syrinx that is producing the sound, and you'll find that neurons in the brain stem that project to the syrinx, so that this is the neuromuscular junction here. These are so-called motor neurons that project to the muscles. Different neurons will project to different muscles. So there is some sort of muscle-based topography in the system. We call it a myotopic organization. It's a type of topographic organization. And if we now do the same experiment, putting injections of tracers into the brainstem nucleus, we'll find that there are different regions within this RA structure I mentioned a moment ago that receive input from the different neurons down here. So the myotopic organization is preserved in RA. Okay? If we now do the experiment in that subdivision of RA, if we now do the same experiment in that subdivision of RA, we find that throughout this other pathway I mentioned, the AFP, that this myotopic organization is maintained. This slide is a bit old because what it claims is that there's no such organization within HVC. That is, topography is broken down there. And it turns out we now know that there's a very complex, but there is a topographic organization in HVC. It's just not a point-to-point connection. So we can learn something about the anatomy of the system from that. And we can also recognize that in RA, there's a group of cells that project down to the pathway that's controlling the muscles of the syrinx. And there's a group of cells that control that projects to the brainstem structures that control breathing. Why is breathing important? Because for instance, as I'm speaking to you now, I have to push air through my glottis. And I have to do it in a regulated fashion. And I have to do it in conjunction with breathing, right? As I remind my students all the time, singing is important, but breathing is more important. So the animals have to do that very well, or they're not going to survive at all. So, and it's in fact these breathing centers in the brainstem that give rise to feedback going up into the forebrain. Okay, so we're going to simplify the problem a little bit and focus on these three structures: HVC, this anterior forebrain pathway, and RA. And if we can learn something about the interactions of those different parts, we can start making some progress, right? So we're going to start by focusing on RA. And let's start by looking at how we collect some of the raw data. So you're now going to see a bird. The bird is going to be in a cage. It's going to be wearing some headgear. It's in a half cage, actually, and you don't see it in the video, but next to the animal, it's a male. In this species, the males sing and the females choose males based on the quality of their songs. So the male bird is singing to the female. trying to impress her with the quality of a song and maybe the fancy chapeau that he's wearing. And you'll hear both the bird singing and the neuron firing. This is a recording of one cell in RA as the bird sings. Okay, so that's the setup. That's the neuron. That's the song. Regular. And you can see how remarkably similar the pattern of activity of the neuron is when the bird sang A, B, C in both times. But after this, he had a pause and then he sang D. And after this, he had a different length pause and he sang E. And prior to singing D or E, the pattern of the neuron already changed. So by that basis, we would argue that this activity is pre-motor. It's leading. The change in the neuron leads the change in behavior. That's interesting. And so that's consistent with this RA structure being a motor cortical structure. Okay.